Computations and calculations are great at a certain stage, but the real test is what happens in real life, what happens in the experiment. The real test of something is, does it allow you to make progress?
How are predictive tools changing the way we discover drugs? And how long has that change been occurring?
When we think of predictive tools, we tend to think of relatively recent times. Terms like machine learning and artificial intelligence seem to have sprung up in the last decade, at least in the popular imagination.
But our attempts to predict good drug candidates goes back farther than that. I'll bet you're familiar with one of those predictive tools, Lipinski's Rule of 5.
I actually do remember that from my undergrad. So how did these first human-usable predictive tools come about?
Hi, I'm Charis.
And I'm Jesse, and we're the hosts of the Analytical Wavelength, brought to you by ACDLabs.
In this episode, we'll discuss predictive tools in chemistry, both past and present. And we'll start the story with none other than Chris Lipinski himself. Here with me today is a scientist who will be familiar to many of our listeners. Chris Lipinski worked for 32 years at Pfizer, where he retired as a senior research fellow. Since then, he's been consulting and advising pharmaceutical companies. He's famous, of course, for, among many other things, Lipinski's Rule of 5. Thanks for joining us today, Dr. Lipinski.
I'm very pleased.
And we're very pleased to have you. So, let's start with our usual icebreaker question. What's your favorite chemical?
That's a very easy question for me to answer. It's a compound I made in 1971 at Pfizer. The USAN name of that compound was tolimidone. And I'm still involved with that compound 50 years later. Tolimidone was CP26154 when I made it at Pfizer. And then after I retired and I joined up with Milieu Discovery on their scientific advisory board, I suggested that compound as the first compound for Milieu's independent drug repurposing efforts. And the people at Milieu discovered that the compound had a spectrum as effects on glucose tolerance, positive effects improving glucose tolerance. Biologically, it was acting like an insulin sensitizer. Milieu people discovered that it had a completely novel mechanism. This was about 2007, maybe. It was a lin kinase activator, an activity that had never been reported before, and it still is the only known lin kinase activator. The compound was taken through phase 2B, and it was successful in meeting its endpoints in treatment of type 2 diabetes. So scientifically, it could have progressed, but the economic analysis of the time left on intellectual property coverage and the competing compounds in the market. So it's basically an economic analysis. And the other thing that makes that compound very unique, it is a compound that was made in the era of before mechanistic screening. The first screens in that compound were actually in dogs. And it's very low molecular weight compound. It's really solidly in the fragment range. And despite that, it has activity at 50 nanomolar as a lin kinase activator. So it's a very, very unusual profile. So anyway, that's my fun compound and my favorite.
Oh wow, that's definitely a good one. And really cool that you've been working with it for decades. So you've been very influential in the field of chemical predictions, particularly related to the Rule of 5. Could you provide our listeners with a brief reminder of what that particular rule is about?
Sure. So this is a rule that came out internally at Pfizer. I came up with it in 1975. It was published, I think it was in November of 77. I'm sorry, not 75, 95. Discovered in 95 and published in 97. So it's based on an analysis of compounds that received a United States adopted name or a British adopted name, which is an indication that the compounds actually were suitable to go into phase 2 clinical studies. So they had gotten through phase 1. And the observation was that there was if you looked at the distribution of compounds by a series of physical-chemical parameters that were known in the literature to be related to, for example, oral absorption and solubility. The distribution of those parameters followed a pattern where approximately 95% of the compounds were in a certain range. 90 or 95% were in a certain range. And so then the rule became a very, very simple one, which was that you are more likely to get a compound with solubility good enough to achieve something orally, or permeability good enough to achieve some kind of activity orally if the molecular weight were less than 500, the number of hydrogen bond donors was 5 or less, the number of hydrogen bond acceptors was 10 or less, or if the calculated log P was 5 or less. And then that rule is actually for the types of compounds that was based on, which were all pre-1995 compounds, has held up very, very well with time. But the problems that existed in 1995 on the solubility front and permeability front, the solutions for solubility came much earlier. They started to come in around the year late 1990s, year 2000. The much more difficult problem of enhancing permeability for difficult compounds, they really have reached their fruition, sort of, I would say, in the last 5 years. So Rule of 5 actually provides a good benchmark of the kinds of compounds that we really like to have in drug discovery if you get them to succeed with the least amount of effort. It is possible to get beyond Rule of 5 compounds and to get them approved, get FDA-approved drugs. But the degree of difficulty is very much more difficult. And the range of compounds that are likely to be successful is less, but it is still possible. And that was the improvement in solubility and in permeability was not predicted in the Rule of 5, because actually a lot of those technologies were either immature at 95, 97 time period, or in the case of permeability, basically at the very, very most nascent stages and very little published literature on them.
Right. So before the Rule of 5 came about, which helped scientists pick those drug candidates which might be most efficient, how were drug candidates prioritized? Did researchers rely on their intuition or something else?
Actually, I would say there is always intuition, and the best scientists actually usually has intuition. But no, drug candidates were chosen based on what were very logical rules at the time, with one major exception, and that major exception was that sort of the late 1990s was the time of high-throughput screening, and there was a tremendous focus on sort of reductionist thinking and a huge focus on just potency, and people forgot about the types of properties that a drug needs apart from potency, that a compound needs apart from potency to actually become a useful drug. And that time period was the time period where the drug discovery was essentially almost wholly dominated by pharmaceutical companies. And academic drug discovery really hadn't really come in at that point in time. So I would say the drug companies, they knew what they were doing, and they had highly well worked out processes for selecting drug candidates. But it was in the era of high throughput screening, which definitely affected the world view about what you needed for a drug candidate. And also it was a time period, the late 1990s, where there tended to be in many organizations, including at Pfizer, the discovery departments would tend to be in one part of the organizational structure, and then the drug metabolism and the pharmaceutical sciences were almost universally in another department, in a development department. And so that also sort of hindered the ability to take into account the more development properties and to bring them backwards into the discovery process.
Mm-hmm. So obviously there were some very systematic processes in place even back then in 95, but there's been a lot of improvement since then as well. You were talking, for example, about our understanding of drug permeability. So how do you think our understanding of what makes a good drug candidate has evolved over the last few decades?
So I would say, the reason Rule of Five came out in 1997 is the timing was perfect. Effectively, my lab at Pfizer, I think, was worldwide the first laboratory in a discovery setting that really went after the measurement of the physical chemical properties in a discovery setting. And that meant that, we had to take some shortcuts. When we measured solubility, we didn't measure thermodynamic solubility. We measured, kinetic solubility, but that didn't matter because our chemists were effectively making almost entirely amorphous compounds. So, the crystalline state on the solubility really doesn't matter. And what we really were trying to do was to influence their choice of compounds. And we were successful at that. We actually rolled the Rule of Five out at Pfizer internally in 1995. And it was moderately successful, but, medicinal chemists are a smart bunch, and a lot of them would say, well, yeah, this is a calculation, and yeah, but I don't really believe too much in calculations. I mean, there was some considerable degree of skepticism. So what we did is that within about a year of the time we rolled it out, we actually came up with discovery kinetic solubility assays that were capable of testing 10,000 compounds a year, which was enough to keep, at that time, was enough to keep up with the medicinal chemistry output. There were some management scientist interactions that helped this process along. So, our chemistry managers knew that we had a problem with. We nominated a clinical candidate, and the poor pharmaceutical sciences people would just throw up their hands and say, "What are we supposed to do with this compound? The properties are so bad." So how do you solve that problem? Well, the first way to do it is maybe try to identify those compounds, maybe by calculation a priori, that really are likely to have, those miserable properties that the pharma sci people can't handle. And that's where the Rule of Five came in. A key decision that was made, and actually I have to give credit to the person who at that time was in charge of our discovery computational chemistry group, who also had control of the compound registration system. I worked with him. His name was Beryl Dominy, and he put the Rule of Five into our registration system. So as soon as the registering the compound, entered it into the registration system, and the compound structure went in the background, immediately all the Rule of Five parameters were calculated. And if there was a problem with one or more of the parameters, they would see it at the time of registration. So the advantage of that was that it completely eliminated any excuses. Well, we never heard of this Rule of Five stuff, and we don't know how to do it. Well, it appeared in the registration, so they couldn't say that. But where the managers came in is that all the chemistry managers found out about this, and they found out they could get a printout or they could see it online. So they could actually track their people in their groups and see, who are the people who are making the more reasonable compounds. I still remember quarterly meetings where, a manager would ask, how is your compound compared with the Rule of Five? And then the medicinal chemist would be on the spot, and they couldn't say, well, I never heard about it, or I don't know. But nevertheless, we still had some reluctance. And then once we rolled out the solubility assay, the discovery solubility assay, then the medicinal chemists were much more comfortable about that, because it was honestly I was so many years a medicinal chemist. Computations and calculations are great at a certain stage, but the real test is what happens in real life, what happens in the experiment. And so now you have an experimental assay. And that's sort of the story of the acceptance of the Rule of Five. And actually, the whole story is published. I had an essay in Annual Reports in Medicinal Chemistry. I think it was 2014. There's a good, almost a page on that, on the actual real story of how we discovered how we came on the Rule of Five. I was working on something that wasn't even on anything that I was supposed to be working on. It was just the summertime, and I was working on a new statistical program from SAS that I got in, and I saw all these graphing possibilities. So it was just the story was that you can get a lot of things done just because they're fun, and also because if you want to come up with things, you have to have time. What kills creativity is a Outlook calendar that's full all the time. So I had all kinds of strategies so I could carve out blocks of time where I wouldn't be bugged by phone calls and worthless meetings and so on. I would really have a chance to think. I mean, there's actually a lot of scholarship on this. Frequently, to come up with ideas, you can't do it in 15 minutes. Take just 15 minutes or 20 minutes just to calm down, and it may take you an hour and a half, of uninterrupted time thinking about something.
Absolutely. It's really amazing what can come out of just playing around, satisfying scientific curiosity, and it's great to hear that the rule of five is actually one of those things. So let's go back to you mentioned that there was a bit of mistrust for people who thought, oh, this is just a calculator, and also that the rule of five works for many things, but that there are exceptions. So how do you feel scientists should balance their own expertise, intuition, and desire to explore alongside the guidelines that these calculators and tools are giving them?
I do have a definitive point on it, and I think the real test of something is, does it allow you to make progress? Does it allow you to be more efficient, or does it allow you to actually succeed? And that's really, to me, more important than something like, the correlation of calculated value versus experimental value, right? I mean, that is important, but the more important thing is the bottom line. And I will say on the rule of five, I had tremendous internal support at Pfizer at that time from the pharmaceutical sciences people. Even though the way I was doing my experimental assay broke every rule that's in the textbook at that time on how you should measure solubility, right? Because we were doing it by detection of precipitation using a diode array UV machine. I mean, it doesn't appear anywhere in a textbook. And it's in a classical way. It's an incorrect way of measuring solubility. But the point is that the pharmaceutical sciences people were tremendously frustrated by the quality of the compounds they were getting from the discovery people. And anybody who could come up with anything, even if it wasn't a classical approach, that could improve things for them so that their job would be easier and they could turn a discovery compound into a compound that the formulation and the characterization that would be successful so it could be passed on to the clinical people to go into phase one. I mean, that's what they wanted. And so what I was doing, even though it broke the rules, it made their job easier and overall made the company more successful. So I actually think there's nothing wrong with being skeptical about calculations, and there's nothing wrong with being skeptical about experiments. So you should be skeptical on both sides. There are undoubtedly errors in calculations. My attitude is a lot of times the exact correspondence of a calculated value to some experimental value is maybe less important than the trend you see, because you want to move medicinal chemistry in the correct direction. So maybe you're off by a factor of 3 or something, but as long as it's a fairly consistent error that you're off, it doesn't matter because you're going to be moving in the correct direction. And then the other thing that's not appreciated is the error that you get in experimental physical chemical measurements. So again, this time frame of the late 1990s, 2001, 2002, I actually worked quite closely with a Pfizer manager. He was a very, very good chemist, and he was in charge of environmental packages on our compounds. And as part of that, we had to get experimental log P measurements, pretty accurate measurements of some pretty high lipophilicity compounds, which is incredibly difficult to do and horribly expensive. I mean, we were spending, and this is back in like 2000, to get a compound whose log P was around 5, to get an acceptable experimental value that we could submit to the EPA. I mean, that would cost us back then, $20,000, $25,000. And we had to, come up with radio-labeled material and show that the partitioning of radio-labeled material into the lipophilic phase was due to the compound itself and not some radioactive impurity, and we had to do it at 2 concentrations. It was really, really difficult. And I still believe today that most of the experimental log P values of, let's say, 5 or higher are probably wrong. And the usual pattern is that those values are far too low than the true value, which is in some ways kind of meaningless because at high log P values, compounds, they're certainly not discrete monomeric compounds. They usually self-aggregate and highly protein bound in biological media and so forth. So I think having a suite of software that you're comfortable with, that you use routinely, that covers a range of properties that are calculated, is extremely useful. The earlier it's used, the higher the value, and then as time goes on, late discovery and certainly into by the time you get into clinical, then the clinical parameters take over. So experiment always trumps calculation, and calculation has its highest value when either the experiment is difficult to do, which can easily be the case with a compound with difficult properties, or when it's just, for example, impossible to do. For example, when you're actually planning out what you're doing, so the compounds don't actually exist. A competent, experienced medicinal chemist always should have a very good sense how their compounds behave, what is responsible for the potential solubility and permeability issues, what can they do in chemistry to try to fix those.
That's really useful advice in terms of keeping the end goal in mind, using all the tools at your disposal, and always just trying to move in the right direction whenever you're designing compounds.
Right, and also, one of the things, though, after I retired, I'm still doing a little bit of it, did an awful lot of legal consulting work and working mostly on patent litigation. One of the things we would always get from the people challenging a patent was to say, well, you guys are reporting activity for a compound, or you're just reporting a IC50, but you're not bracketing it with 95% confidence limits. And that was very typical for chemistry publications. And the journals allowed it. And there's a reason for it. So a biology experiment typically it's a group of animals, and you're getting some value out. But it's essentially a single compound against a single test, maybe repeated enough times. But that's very, very different than when you're doing medicinal chemistry SAR, when you're looking at the pattern of activity of a compound. And so a single point is much stronger. Okay, there might be an error there, and you don't know about it, but you'll pick it out in the pattern of compounds because, you go along and you say, wow, this compound's active. Well, why wasn't that compound back there, active? Then you go back and retest it. Oh, yeah, it really isn't active now. So the errors tend to self-correct. And medicinal chemistry lab is always faced with the decision of what compound I'm going to make next, and I'm going to have to balance all of the oftentimes competing data on the compound, so balance potency versus solubility and permeability, and metabolic stability, and whatever standard assays you have that would eliminate a compound, you've got to pass those. So it's very often, in fact, most of the time, the compound that gets nominated is the compound that is the compromise. It's got the best pattern of activity against those parameters that you think are important. But against any one parameter, it may not be the best compound, but it's the best set.
Definitely a lot of things to keep in mind here. You've shared a lot of great experience and stories with us today. What do you think is the most important advice for our listeners to take away?
You have to understand your compound. You have to understand what its properties are. And when you're thinking about making something or planning something out, you need to know, whatever compounds you're planning, how you might predict that they might behave. The kind of thing that was done in pre-Rule of 5, which was to solely focus on potency, is just at least for drug discovery, true drug discovery, is a total loser. And that, by the way, is the reason why the Rule of 5 took off. From the time it was published in 97, it just every year the number of references to have increased. Now there's like approaching 21,000 references on it, and this year it's already more than it was last year, which is more than the year before. So and there's a reason for it, which is that it's useful. And the other thing that's very useful about it is it's simple. It's very simple. Actually, it's been criticized as being too simple. But you don't need a computer program. You just look at the structure, and except for the log P, which you do need to have some a calculation for it. But the other parameters, you can get them right from the chemical structure. It's not a problem.
Thank you very much for your contributions to medicinal chemistry, of course, but also here today on our podcast. It was a pleasure to have you.
Okay, bye-bye.
Take care. Dr. Lipinski gave us a wonderful history of what predictions used to look like and how he came up with the Rule of 5 that's still famous within the industry.
Of course, chemical property prediction is still important these days, and if you want to learn more about that, you should check out the Perceptive products on the ACDLabs site. You can find a link for that in the show notes. But there are new fronts in the efforts for computer-assisted drug discovery.
To hear about current advances in machine learning for drug discovery, we turn to Dr. Olivier Elemento. Here with us today, we have Dr. Olivier Elemento, who is director of the Englander Institute for Precision Medicine, associate director of the Institute for Computational Biomedicine, and a professor of physiology and biophysics at Weill Cornell Medicine. He has published extensively on the use of AI and machine learning in topics related to drug discovery, and we're really excited to have him here today. Welcome, Olivier.
Thank you so much for having me.
Very excited to talk to you today. I want to start off, though, with our regular icebreaker question. What's your favorite chemical?
Well, that's a great question. I don't know if I have one favorite chemical, but I've always been very impressed with natural products, drugs that come out of natural products. I'm always very impressed with, the amazing sort of tricks and the amazing creativity that, millions of years of evolution have created. I think it's just amazing to see these, natural products. We do a lot of work in cancer, and a lot of cancer drugs are coming from natural products. And, it's crazy to see the complexity of these molecules that, essentially have been designed by, millions of years of evolution. It'd be possible probably for human beings to design them from scratch. They kind of evolved, and as a way to enable organisms to kind of, defend themselves and fight each other and so on. And it's just, amazing how complex these molecules can be. And we're obviously very interested in getting some inspiration from the complexity of these molecules.
Yeah, definitely. Nature is an amazing designer of molecules.
Yeah.
Yeah. So moving on to our main topic of today, how did you get into this sort of intersection between AI and drug discovery? Did you start as a computational scientist, or did you start on the biology and science side?
Well, thank you so much for asking this question. So my background is initially engineering. That's how I started my studies. I was always very interested in computer science and was also interested in biology. And I was actually very interested in the application of computer science to biology. And that's why I did a PhD in computational biology. And it was very rewarding. I really enjoyed it very much. Sort of later on, I joined Cornell and started working on our program focused on precision medicine here at Cornell. And, that program involves sequencing a lot of genomes, especially cancer genomes. We've been focusing on trying to understand what's driving individual cancers, the mutations that make each cancer unique, and especially the actionable mutations, what we call actionable mutations, which is the mutations for which there is potentially a drug that can target the product of the mutations. And there's a growing number of such mutations, as you know. That all being said, as we've been sequencing genomes, we also have realized that there's a very large number of mutations that are not actionable. Very often you don't understand what these mutations are doing. There's no drug that targets these mutations. Sometimes you actually start understanding the function of these mutations as you do experiments. You can model these mutations in a variety of different ways. Using mouse models or cell models, and was trying to understand them better. But nonetheless, there's no existing drug that can target the product of these mutations. And that's really actually, for me, it was almost a sort of aha moment, in a sense that we sort of started realizing that maybe there's potential to use AI as a way to create new drugs that could potentially target these mutations whose function we just did not understand, or whose presence we could not act upon when we see them. And so that's what started a lot of the ideas in my lab in terms of using AI as a way to develop new drugs and discover new drugs. And there's a lot that we've done in the past on this, a lot more that we can do in the future. I think it's a very interesting space. There's a lot of potential to use AI and data science as a way to transform how we develop drugs. And a lot of my focus, a lot of our group and our institute has been focused on these kind of problems.
Great. That's very fascinating. Now, I wanted to talk specifically about how you see AI as solving these problems. People can say like, oh, just throw AI at things and it'll solve it. But so why do you think that AI is a good approach for solving these drug discovery problems, and that'll be helpful for developing drugs for the targets that you're thinking of?
Yeah, so we are using AI in lots of different ways, trying to address complex, difficult problems in drug discovery using AI. One of them, just to give you an example, and that's something that we worked on early on, was trying to predict ahead of time if a molecule is going to be toxic in human beings. So right now, as you know, this is not a simple problem to solve, right? It's typically addressed using mouse testing or animal testing, a different kind. And even so, once you have molecules that do not seem to be toxic in animals, as we know, many of these molecules are nonetheless toxic in humans. They failed phase 1 trials, for example, or even fail later. So we've been trying to see, for example, if we could address this kind of problem using a data science and AI approach. A few years ago, what we did was to compile a very long list of molecules that failed in clinical trials because of toxicity reasons. We also made a very long list of molecules that did not fail, that went all the way to approval. And then we asked essentially, can we learn something from these molecules? Can we learn something from the molecules that failed versus the ones that did not fail, that was eventually got approved? Can we describe the molecules using different features of these molecules? Can we describe the molecules in terms of what they target in human cells? Can we describe the targets of these molecules, what they interact with in cells, where they are expressed in different cells in the body? We made a list of features for each of the molecules. And then essentially we used a machine learning method to predict, or to try to predict, which molecules failed and which molecules succeeded in the drug discovery process. And as it turns out, this very simple approach, right, gave rise to a model, a predictive model that was actually quite good at predicting which molecules failed in clinical trials, in phase 1 trials. We've validated these predictive models in different ways, but it just gives you a sense of the power of AI to kind of learn from all the data that we have access to address very specific questions. I think part of the reason why there's a bit of hype in the field, and maybe AI has not delivered, if you want, as much as some people thought it could, is because sometimes you hear, like you say, that AI is being applied in general. What we really believe in is really application of AI to specific questions, specific problems. Can we predict if a drug is going to be toxic or not? That's very specific. There is data that exists. There are labels. That's something that's important in AI. You need to have specific things to predict. We call them labels, like it's toxic or not toxic. Very simple label in some ways. And then you can describe the drugs using all the features that I mentioned. That's essentially a well-formed machine learning problem. We thought that maybe it could work. We took some inspirations from what's happening in other fields. For example, you may have seen this movie called Moneyball with Brad Pitt, where it's coming from the book. But essentially, it's the application of data science to predicting the extent to which baseball players would be successful in the future. It's exactly the same approach that I described. You take a lot of players who've been successful, a lot of players who haven't. You describe them using different sort of features, and then you try to predict the future. And it worked in baseball. We did the same thing for drugs, and it worked, as well. So I think there's something to be said about this kind of approach.
Absolutely. So you talked a bit about the list of features that you need in order to build your model. And I think one of the challenges in some fields, at least, is getting the data to feed these models. So where do you look for the source of data for these features, and how long does it take you to train one of these models?
Yeah, so in this particular problem that I mentioned, the problem of predicting toxicity of molecules, there's a great source of data, which is the very large number of clinical trials that failed and the trials that succeeded and that sort of moved into phase 3 and then eventually approval. So there's a website for this called clinicaltrials.gov, and then once you have information about which drugs failed or not, then there's an additional database, for example, PubChem or Drug Bank, that have information about the molecules and information about how to describe the molecules or to describe the targets of the molecules and so on. So there's a lot of data that sort of exists in the public domain. One of the challenges of the field, and that's something that my lab is really trying to specialize in, is data integration. Because I think the challenge that we see across the board is that there's a lot of data, but it's coming from different databases and it's often not connected. So our lab has made a lot of effort to try to build a machinery to be able to connect different pieces of data together so that we can sort of address complex questions like the ones that that I discussed with you. But we've applied similar types of machine learning, for example, to predict what's the target of a given molecule, right? A lot of molecules come out of phenotypic screens, as we know that they do something interesting, but we don't necessarily know how they do so. We've been thinking that we could potentially use machine learning as a way to predict the target of a molecule. And the same thing, right? There's a lot of known molecules with known targets. Use that information as a way to train how to connect a molecule to its target based on features of the molecule. And likewise, the secret for this was data integration. Was to realize that there are different ways to describe a molecule. You can use, for example, what happens to cells. If you treat cells with a molecule, you see that there are genes whose expression goes up and down. That's information about the target, but it's not the whole information. You can treat, let's say, 100 cell lines with the same molecule. Some cell lines will die. Some cell lines, will just, essentially not care about the treatment. It's like, a profile of cell killing. That also has information about a target. It doesn't tell you the target, but it's an additional piece of information. You can have side effect information. You have a structure of the molecule itself. In that project, what we did was to integrate all these pieces of data together. We basically say, look, each one of them gives us a little signal about what this molecule is doing. What about if we integrate all this information together? And as it turns out, we saw very clearly that the integration of these data types led us to very good performances when it comes to predicting the target of a particular molecule. I think this is something where we learned that data integration is key. It's not easy, kind of like you said, the challenge is to compile data that's connected, to each other. Databases are not typically connected, so we have to work to connect them. As I said, we built a platform for doing this. But once you do, I think you have a very powerful data set that you can use to address all kinds of different questions.
Lovely. And I love hearing all of these examples that you've shared from your research. I was wondering, though, if there was any other examples of things that you've done that were particularly exciting achievements from the application of AI that you could share with the audience.
Yeah, absolutely. Plenty of projects. One of the projects that I think has been really exciting to me and to us, here at Cornell, is to be able to analyze pathology slides for interesting signals. So as you may know, every time that somebody is diagnosed with cancer, the diagnosis is coming from a piece of tissue from the cancer that's stained with specific dyes, with the idea of being able to understand the architecture of a tissue, the morphology of cells, and in the tissue that's suspected to be cancer. And so from that kind of tissue analysis, one can make a diagnosis, a precise diagnosis about the type of cancer. These tissue sections are very often digitized, and so they exist now by the millions. There are millions of digitized pathology slides that exist in pathology databases. There are companies that now have really enhanced the field when it comes to digital pathology. The point is that there's a lot of data that exists. One thing that we did, it was published a few months ago, was to try to see if we could extract information about these slides for example, to tell us what kind of mutational process is happening within the tissue, within the cancer. We essentially compiled a very large database of pathology slides for which we also have, we had genomic information. We had information that could tell us whether a mutational process was happening in some of his tumors and not others. And using AI, we were able to connect the images of the tissue to the mutational process that was happening, inside the tissue based on genomic information. And we're able to make this automatic connection. And now the idea is that we built a model that can just take H&E slides, these pathology slides, and is able to tell without actually having to do genomic analysis if the mutational process is active or not. So I think that's a great example of how we can potentially get a lot of useful information out of pathology slides, including potentially about mutational process, some of which are potentially targetable by drugs, that could be used maybe as a way to stratify patients, find patients that have a particular profile. Maybe these patients will respond better to a particular drug.
So those are some really great successes. But I also wanted to ask about the flip side a bit. What do you think are the greatest challenges right now to making AI a more productive part of the drug discovery process?
Well, I think part of the challenge is that AI is good at interpolating. It's good at sort of learning from the labels that, we have. It's good at learning from the data that we have. It's not always good at extrapolating, right? So we were, for example, discussing earlier molecules, natural products, right? These are really complex molecules. If you look at the drugs that we've designed, that human beings have designed, or that came out of different types of screens, or sort of targeted screens, they all kind of fall into specific scaffolds, specific categories. It's very hard for AI to learn from existing data and kind of think about the universe of drugs that is outside the universe that it learned from, right? So AI is good at interpolating. It's not great yet at extrapolating. And I think that's a bit of a challenge. I think this is something that we need to work on as a field, is sort of understanding better how to extrapolate and not just focus on the universe on which we're learning our AI models. That's a challenge to some extent. The other challenge that I think we see, and that maybe was applicable to this pathology AI project that I mentioned, is that we're able to make these great connections between, let's say, images and specific processes, let's say a mutational process, and so now we can essentially apply an AI model to a tissue section, and it tells you a patient has this mutational process, for example. But by and large, the AI models that we create, can't really sort of explain themselves very well. We have a limited ability to understand precisely in in terminology that we can understand and communicate with each other about what's happening within these AI models. And I think that's something we need to address as a community. We need to make these AI models more explainable. And I know that some people are working on different techniques to do this. One of them is this class activation maps, right? Where basically what you're doing is to take an image and ask the machine learning model to tell you the pixels where it's looking in the images. And that's okay, that's a good starting point, but it's not, I don't see it as a great starting point for a discussion, right? Because if you show a bunch of pixels to a human being, it's hard to have a discussion about what these pixels are. Like, it's not as information that's useful when it comes to, for example, a physician explaining what a machine learning model is doing. You can say, well, there's a bunch of pixels that are blue, and that's not going to help much in terms of interpretation. So I do think that we need to come up with better ways to interpret models. This is something that the community is working on. I think there's a lot of work to do there, but I think this is a limitation.
Excellent. I think that's a really interesting summary of some of the questions that AI is facing right now. Are there any other final thoughts that you wanted to share with us around the trajectory of AI or where you think things are going in the future?
I think there's a lot of potential in this field. I always say that I really believe that the way we develop drugs, I think is going to be completely changed using data science. There's a lot of data that we can learn from. I think the drug industry is starting to embrace this idea that you can learn from existing data and then use that data as a way to do better, sort of be faster, fail less often, There's amazing potential. I do think, as I say, that the key is going to be to address specific problems. There's a lot of also interesting work to be done, I think, in my view, for example, to understand where we have data gaps. Because, a lot of us are kind of working on sort of the data that already exists, and I think we need to kind of map out the universe of, certain things that we measure, for example, drug structures, understand where there are gaps. And, I think there's going to be a lot of really interesting, maybe things to discover in those areas. We haven't gone there yet, and I think this is important. We shouldn't just be satisfied with the data that we have. We need to think about what data we need in the future. And I think that's also something that is maybe not told as often as I would like it to be told. I think there is really great potential to better understand the data universe and kind of fill the gaps once we understand that universe.
Mm-hmm. And I think that's a really great message to take away for scientists who aren't necessarily working in machine learning, but there's data that they can contribute to it as well.
Absolutely.
Yeah. Well, thank you so much, Olivier, for joining us today. It was great to have you.
Thank you so much for having me. It's been a pleasure.
Thank you for joining us.
All right. Take care.
From the rule of 5 to the latest in machine learning, this episode has taken us on a tour through the development of predictions in drug discovery.
It's been quite the journey, and it's the perfect conclusion to our season, which has been all about predictions, how they work, and how they complement scientists' work.
We hope you've enjoyed following along with us on this journey.
Jesse will be returning next year for season 3. This is my last season hosting the Analytical Wavelength, but I'll definitely be tuning in for future episodes. If you want to join me, remember to hit subscribe.
Keep an eye out for some bonus content that might be dropping in the feed between seasons.
Thanks everyone for joining us on the Analytical Wavelength. The Analytical Wavelength is brought to you by ACDLabs. We create software to help scientists make the most of their analytical data by predicting molecular properties and by organizing and analyzing their experimental results. To learn more, please visit us at www.acdlabs.com.